Hey guys, so it's Rhiannon and Lauren here, and we're going to be going over our gene presentation and how using genes found in Arabidopsis plants was used to examine space travel and lots of other cool little things.
So, recent interest in growing plants in space has emerged due to potential advances in biotechnology and medicine, with particular focus on biomanufacturing, gene expression, and gravitropism. Our research addresses these topics by utilizing databases such as BLAST and Thalamine to investigate the presence of specific genes in various Arabidopsis plants. Along with analyzing volcanoplots, we also use CRISPR-Cas9 technology to enable precise gene editing, allowing us to target and modify specific genes to achieve desired outcomes. The genes identified in space datasets can be used to explore how plants adapt to the microgravity conditions necessary for survival in space. Additionally, studying the evolutionary history of these genes provides insights into their function and potential impact. Of particular interest are the gravitropism trends associated with our gene of focus, which may have significant implications for the successful cultivation of plants in space environments. While being enrolled within genetics lecture and lab, we have spent a large portion of our lab researching this topic, selecting our gene, and incorporating metagenomics into our research. Today in our presentation, Rhiannon and I will be discussing our research of gene AT2G36590 and its evolutionary history, role in spaceflight, and methods used to incorporate CRISPR. So, over our Venn diagram of spaceflight, within our study, we began by using RNA sequencing data to assess the activity levels of our target genes. This enabled us to identify which genes were most expressed under specific conditions. We then focused on Arabidopsis data using databases and examining the differential expression of genes between the root and leaf tissues from the databases. By analyzing the Venn diagram and comparing the expression profiles between these two tissues, we identified our gene as a gene of interest, the AT2G36590. Based on our analysis, we hypothesized that this gene could play a crucial role in spaceflight, potentially contributing to plant growth and adaptation under microgravity conditions. 210 genes were found within the root tissue and 324 genes were found within the leaf tissue, leaving 16 genes to be overlapped, leading to our hypothesis as to why we assumed this gene would be beneficial. The amount of gene overlap between root and leaf supports our idea of the gene being involved in spaceflight. And then moving on to gene expression analysis in spaceflight using two gene databases representing the root or the purple and the yellow in the leaf tissues were analyzed to investigate the expression of our gene AT2G36590 during spaceflight. Log twofold changes and the p-values were calculated for both datasets. In the yellow leaf and the purple root databases, the log 2 FC values were negative, indicating our gene was not expressed or was downregulated in spaceflight conditions. However, the root dataset showed that the gene was statistically significant, which could suggest a response to spaceflight. This is overexpressed as well. These results provide insights into the differential expression of our gene in response to spaceflight conditions in different plant tissues.
Okay, so now we're going to be talking about some of our goals that we found through our research. So several branches of our gene's origin was researched. These included which Arabidopsis plant our gene was found in, if our gene was used in space travel, what the developmental transcriptome was, and its evolutionary history. Our research focused on investigating multiple aspects of our origins and characteristics of our gene. We explored its presence across Arabidopsis plants and examined whether it had been utilized in space travel studies or not, and analyzed its developmental transcriptome, and we also traced its evolutionary history. Each of these branches provided valuable insights into our gene's functional roles and potential applications, contributing to a deeper understanding of its biological significance. So we found that in our gene, we had a total of 2 transcripts, 13 exons, and 11 introns. Our gene of interest was found in Arabidopsis plants with an ID of 3702. The gene also goes by the name proline transporter 3 and has a secondary identifier of locus 2044835. The length of our gene is 2,668 and it's located on chromosome 2. So moving on to what we did with our gene and some of the methods. First, we made a CRISPR guide targeting our gene of interest. CRISPR-Cas9 is a short RNA sequence designed to specifically target a desired gene within the genome, directing the CRISPR-Cas9 enzyme complex to that precise location to make edits cutting the DNA at that site. So on our poster, you can see our CRISPR guide selection, and that is inserted into, or in between the base BSA1 enzymes. So moving on. Second, we extracted our Casper plasmid from the DNA using E. coli with the QIAGEN mini prep kit. Once we extracted it, we had to linearize the DNA with the restriction enzyme BSA1, which what I was talking about earlier. From here, a gel was run and the larger strand of DNA was extracted. Next, we researched a few mutant genes in the Arabidopsis genome and grew them on agar plates. As you can see on the poster, that's a picture of one of the agar plates that we used. And then finally, nanopore sequencing was done on the Arabidopsis DNA for both plants in microgravity and on ground. And from there, we were able to determine that the number of mitochondria that was required for each of the plants to grow. We sequenced the plants on a clinostat and then used this data to find the mitochondria per cell based on the genome coverage on the mitochondria and chromosome 5. We determined that the plants experiencing microgravity required more mitochondria per cell than the plants did on the ground. This means that plants grown in microgravity require a little bit more energy. Moving on to our gene tree in the corner there. A gene tree is used to demonstrate the evolutionary relationship between a collection of genes and how they have evolved. For our gene, it was found in the mustard family with 9 homologs, flowering plants with 97 homologs, and Arabidopsis plants with 2 homologs.
Okay, and then our next little thing we have on the picture is our little Arabidopsis atlas plant map, which basically just shows us in various plants how present our gene is. So like I said, it shows the developmental transcriptome of A. thaliana based on RNA sequencing profiling. The data set includes the heat stress data from leaf tissue, which can also be used to study the role of stress in splice variant abundance. And also, a little side note, you can use PCR techniques to quantify these variants. And as you can see, areas that are red typically have more of the gene present, and those that are yellow have a little bit more as well, and a little bit less too for the grays. And then gravitropism, we have a little image here. It is a process in which plants respond to gravity. After researching our genes of interest as a class, each group was assigned a gene that was involved in spaceflight, and ours was the AT2G16640. Throughout our research, we found that this gene plays a role as well in the production and gravitropic responses.
And then some future directions that we found. A step that could be taken after our experiment would be which could be related to space-based synthetic biology by designing new organisms. When designing these new organisms using our gene that was present in spaceflight, the new models could be engineered to thrive in space. Another step that could be taken to further our research is the aid in developing crops within space, more significantly gravity-resistant instances.
So as we finish up, we just want to take a moment to thank Dr. O'Connor so much for his guidance throughout this entire genetics course and lab, as well as our research project. We also want to thank him and Kentucky Wesleyan for letting us use their materials and equipment within the lab, especially in pre-professional positions. It's so important to gain research, so we're very, very thankful.
Thank you.
Bye.